So let's just get started pretty quickly. Obviously, special senses. Special senses. I don't know what's going on in the computer. Special senses, as in, photoreceptors of the eye, so you can detect, obviously, light and you can see. You've done senses in terms of proprioception, in neuroscience, in terms of spindle bodies and Golgi apparatus and all of that kind of thing. This one is special senses in the sense that it's chemically mediated. In other words, you've got a chemical that needs to bind to a receptor and then you can detect it. Okay? So the two special senses that actually use this is taste and olfaction, or gustation. That's the fancy word for taste, and olfaction, which is smell. Okay? Now there are other senses like pheromones, but this is not something that kind of applies to human beings, at least I think so. But animals have pheromones, where the animal will go and wee on the tree, and then if the male comes in, they'll have a god-awful fight and all that story. I hope that doesn't apply to human beings. So it may just as well, I don't know. But when I come to something called the vomeronasal organ, I'll come back to that. So, again, chemicals, because obviously the eye is light, the ear is, sound waves, and gustation and olfaction will be chemical senses. There's a strong connection, obviously, between what we eat, what we like, what we don't like. There's also a very strong connection between how we learn with our special senses, and above all, it regulates these kind of functions in our body. So if you're bleeding to death, you have a craving for salt, hunger, emotion. Hunger or hypoglycemia, you can actually start sweating and so on and so forth. Emotion, if you really dislike something, it can affect you for the rest of your life. Sex, as in obviously mating, and forms of memory, where you keep it in your mind. You actually learn from it to the extent where you use neurons and you keep it in your mind that thing really offends you. Again, maybe sweetness has to do with the fact that we are all fed by our moms when we were young, breastfed, and lactose, which is sweet, so we have a fondness for sweetness. On the other hand, medications or things that are very harmful are also very bitter. So there is a tendency to want to kind of take it out of your mouth. So therefore, alkaloids tend to be bitter, and therefore, you do not want to kind of, move it beyond your tongue or your mouth. We tend to like it when we get used to it, like caffeine. Okay, and then lastly, I did the last point up there for you. So if you are looking, by the way, I am not, the lady here is actually drinking Saudi champagne. So there are receptors on the tongue. So, if anything gets into your mouth, these taste cells. So if you are looking at the diagram, I can just get a pointer here quickly. If you're looking at the diagram, there's actually like an epithelial-like cell on the tongue, okay? It's called the gustatory receptor, or the taste receptor. And then between that cell and the neuron, or the gustatory, or the axon of the gustatory neuron, there is a neurotransmitter that would be released. In other words, the actual thing that's determining the taste is not the neuron. That's what I'm getting at, okay? So, again, if the taste sensation gets onto these cells, that will then trigger a depolarizing wave on the cell. That depolarizing wave will then release a neurotransmitter at the synapse between the cell, or the taste cell, and the neuron, and then you will have activation of, obviously, action potentials on the neuron itself. And then that will make its way from the tongue to the nucleus solitarius, and then eventually to the brain. So there is a synapse between the relay of the cell and the neuron. So this would be the depolarization. Obviously, in the cell itself, you will not get an action potential. You'll just get a depolarizing wave, which just means you go from resting membrane slightly up. But if that goes above the threshold in the cell, now you get action potentials, obviously. And then if you remove the taste somewhere here, then the whole thing goes back to normal again. Okay, so taste, there are endless numbers of chemicals that we could possibly taste. However, we can divide our taste into what used to be 4, but now, you know, with the advent of sushi, apparently, we now have 5. So, saltiness, sourness. Saltiness obviously is NaCl. Sourness is H+, or acid, if you want. Sweetness, we know that, sucrose. Bitterness is any alkaloid that kind of tastes horrible on the tongue, and so on and so forth. And then the new one is obviously umami, which is supposedly a response to glutamate. The neurotransmitter glutamate, but in this case, it is the tasted, or the molecule that will give you this glutamate taste, or umami as it's called. There are some substances that actually taste a lot more stronger than the ones we know. So for example, sugars or sucrose is perhaps the least sweet among some of these. Sweeteners like stevia and so on can be extremely strong. And there can be proteins that can be 10 to 100,000 times a lot more sweeter than sucrose. By the way, if any one of you has a formula to convert sugar or amount of sugar to the amount of stevia, please let me know. I've been trying to find one out for quite a while. Never found one. Bitter substances, on the other hand, you require very tiny amounts of them to kind of trigger the bitter taste. And that probably is a protective response, because most poisons or harmful toxins tend to be bitter, okay? So they work in kind of nanomolar kind of quantities. The organ of taste obviously is the tongue. It's not only the tongue, because the back of the mouth, the palate, the uvula, and so on can also taste. But if you're looking at the tongue, there's the tongue, and you have a distribution of these papilla on the tongue. So there's, you have the velate papilla on the back, and then folate, fungiform, and filiform in front. Only the filiform does not have taste receptors, or taste buds if you want. All the other 3 has some version of a taste sensation or a taste bud. There's a histology picture if you want. You've got the taste bud here. There's the taste bud in the form of a diagram, and now you have the taste cells on the inside. So the pink thing is the taste cell, and it gives these microvilli kind of thing at the pore of the taste bud, and that's where you detect the taste. And then at the base here, you will have that synapse between the taste cell and the axon of the gustatory, the neuron that's going to take the sensory neuron to the brain. Again, you have other cells within the taste bud. They will regenerate these taste cells, and I'll look at those in a second. Again, the organ of taste is the tongue, and the way this used to be done a long time ago is you'd get a tongue, and then people would say you taste bitter at the back, sour on the side, salty on the front side if you want, and sweet in the front. That's not the case. That's not the case. Okay? This gives the impression that those taste buds I'm talking about that detect sweet, sour, salty, bitter, and umami are actually distributed in the tongue in this kind of strict kind of way. That's not the case. Okay? So, even though we can taste sweet, sour, salty, and so on and so forth, there is no one single taste that's restricted to a single area. Okay, so yes, you're predominantly sweet in the front, but you can also do bitter and other things in the front as well. So the surface of the tongue, according to the previous slide, have these papilla, and then the shapes you will do in histology. The ridges one, the pimple one, and the mushroom kind of one, the fungiform papillae. And on the side of them, you would have the taste buds. So the taste buds can contain these taste receptor or these epithelial taste receptor cells, and you can have anything between 50 and 150 cells per taste bud. A typical person will have around 3,000 taste buds. Can they be renewed? Yes, they're renewed. They can be reformed every. I think it's on the next slide. About 2 weeks or so. Obviously, if you decide to burn your tongue with hot coffee, it can be reformed, in time, or when you have an infection, like a respiratory tract infection, and then you don't taste your tongue and so on and so forth. So just be careful about this supposed distribution of, specific taste buds in a specific area. Okay, so there's these papilla again. So there's the papilla that's shown here, and you have various kinds of cells associated with it. Basal cells will give you, through cell division, more of these taste cells. Okay, and again, there's the pore, and that whole thing is the taste bud. You have these microvilli in the front that kind of increase the area, if you want, to receive the chemical that will excite that particular taste bud. Obviously, the taste bud is exposed, or the tongue and the taste bud is exposed to the contents of the mouth, and that is what then will activate the taste cells, and then it is the neuron at the base of the taste cell that will give you the sensation that will go to your brain. So there's a synapse between the taste receptor or the taste cell and the gustatory axon that's attached to it. So you're not detecting it directly on the axon. In the nose or olfaction, you're detecting it directly on the axon. There we are. The lifespan of a taste cell is about 2 weeks. Okay, and if for any reason the axon to the taste bud is cut off, that's when you will lose the taste. So there seems to be this kind of relationship between the axon that allows the taste bud to regenerate itself. Okay, I've shown a diagrammatic representation of a taste bud, and I'd like you to live with me for a minute here. So I've got a taste bud, there's a taste bud, there's the pore up here, there's the axon attached to it. Obviously, this is the epithelial-like cell that will detect the taste. And now I've got cell number 1, number 2, number 3. Taste cell number 1, number 2, number 3. And what I'm doing here is are you opening up the one on Moodle or? Never mind.
It's kind of different.
Huh?
Yeah,
It's kind of different. What is
Give
This is the same.
me a second, please. There we are. Okay, sorry. Okay, there we go. There's the lady drinking Saudi champagne. And again, that slide we did. Again, be careful about that one. Then we got that one. And then I inserted this thing with the papilla. All right. Again, chemical sensations, or the chemical will attach to a receptor on the taste cell. Okay, and that will activate or move the resting membrane potential up a little bit, and then once that is high enough, then you will get a neurotransmitter that release between the actual taste cell and the neuron at the bottom. So the neurotransmitter for sour and salty is serotonin, whereas for sweet, umami, and bitter, it's actually ATP. It's a purogenic receptor on the gustatory axon. Okay? Now, in both cases, the neurotransmitter is excitatory. Okay, and that's where I came to this one. Okay, so have a look at this for a second. So you've got the cell number 1. You've got the cell number 1, number 2, and number 3. Now, what this is showing you is the membrane voltage change in cell number 1, 2, and 3. On the top here, these are the tastes that we are going to test. So, if you put salty, and if you look at cell number 1, it gives you a bit of a change in voltage, but then it does that both for salty and for sour. In other words, H plus and Na plus. In other words, cell number 1 is actually detecting 2 different tastes, if you want. Is that okay? All right. Look at cell number 2, it's actually detecting 3. All right? It's doing salty a little bit, it's doing sour really strongly, and it's barely going up for bitter. And then if you look at cell number 3, it's only doing 1.
Sweet.
It's only doing sweet. Now, again, that's the membrane, the voltage, the resting membrane changing from resting, going slightly up. It's depolarizing. Now, look at the effect of this change in the resting membrane potential on the axon. Now, look at cell number 1. Now, notice they all have like a basal level of firing, which is like this, okay? And then the minute they're activated by the particular taste. So if you're looking at cell number 1, notice you have a lot of firing for NaCl, which is why this big thing gives you a lot of firing, a lot of spikes. And then you've got a small one here, and you've got a little bit of spikes there. On the other hand, if you look at cell number 2 axon, you've got a lot of firing for sodium chloride or salty, you've got some firing for H plus or the sour, and you've got a little bit of, slightly higher than basal firing for the bitter. On the other hand, you just got basal level of firing for sweet in cell number 3, and then it goes very high when you put the sucrose in. In other words, what I'm trying to get at here is those cells do not detect one single taste chemical, and when they fire, the firing pattern or the spikes can vary depending upon which taste chemical is activating that particular cell. Okay? Now, I want you to keep that at the back of your mind, because I can represent this in the form of a graph now. And if you look at this graph, okay, you notice red. Can you see red? Yes? Let's say that is sucrose. So that is like cell number 3. It's giving you a high amount of firing, but nothing else for the other ones. On the other hand, if you look at the purple one, can you see the purple one? Now, let's do the green one. The green one is like cell number 2. It's doing nothing for sucrose, it's doing something for salty, maybe a little bit less for sour, and then it's doing something for bitter. You follow what I'm getting at? Okay? Now, the question I ask is this: why does one cell respond to one single chemical, in other words, sucrose 1, cell number 3, but another cell can respond to a variety? Now, and that has to do with signal transduction. Okay? Because what's happening here is some of these chemicals, like sodium chloride, can go directly into a cell. Hydrogen ions can go directly into a cell. That means you can have activation of a single cell with both of these chemicals, yet one is salty and the other one is sour. Okay? Then, the other thing that can happen is you can block G protein coupled receptors or channels. Okay? So let's just look at this bottom end in a little bit more detail. And how do I do that? I'm looking at the taste cell now. There's a cell. I'm looking at salty. So this is the mouth, or the fluid in the mouth. The saltiness, the sodium comes in. You're going to get a depolarization of the cell. the membrane potential is going to change. That's going to open up a calcium channel. The calcium channel will then allow the vesicles containing the serotonin to release the serotonin, and that's going to be excitatory on this gustatory axon. Are we okay? Good. On the other hand, if you look at sour, which is again working through a channel. So there's sour, H plus comes in. What does it do? It inhibits a potassium channel. Potassium doesn't leave. Again, you will change the membrane potential. Again, sodium and calcium will come in through these channels. You will release serotonin. Okay? Now, because both of these are kind of working through channels, you can have this kind of double, if you want, activation of this particular taste receptor cell. Look at the other side now. Bitter, sweet, and umami does not use channels. It uses membrane proteins. They are called T, capital T, and R proteins. And depending upon what combination there is of those proteins, they will then activate a phospholipase C, which will give you calcium. Calcium will now open up a sodium channel, and now that's sodium coming in will allow ATP to leave, and ATP is excitatory on the gustatory axon. Okay? So now you can see you can do more than one neurotransmitter, that kind of thing. And that's why you have this difference in the firing of the axon. So I summarized the molecular mechanism for you. In that previous one that I was using, I wrote it down, and when I was reading it, I decided to make it easier. So sweet. Remember, sweet, umami, and bitter, they use the G protein, they use the T and R proteins. There's a T and R is a group of, it should be taste receptor proteins. And then T1R2 and T1R3 is for sweet, and they use G protein. Obviously, they're activated by things like sucrose and sugar and so on and so forth. Umami uses this group of proteins, again, same as sweet, in other words, G protein, and activated by glutamate. Bitter, again, same as sweet, activated by toxins and quinine and caffeine. On the other hand, salty is activated by an epithelial sodium channel transporter, which causes depolarization, and this one is sensitive to amiloride. Amiloride is a diuretic. Okay? Sour, on the other hand, uses a proton channel, a hydrogen ion channel. Again, increase in hydrogen ion suppresses a potassium channel, causing depolarization, and this is caused by acid, or H+. Okay, so you're getting this activation of the gustatory axon. What happens to it? So again, this is on the tongue, and you know a lot of this because you did cranial nerves already. Okay, so cranial nerve number 7 innervates. Okay, innervates the anterior 2/3, and then you've got the tongue, or cranial nerve number 9 will do the posterior, and then the back will be done through vagus. Good. So you know this. And then once they come through their ganglion, they will go to nucleus solitaris. Okay, and then they make the way to the thalamus and then to the gustatory cortex in the frontal operculum. Okay, and that's your pathway now to the brain. Again, I just summarized that quickly for you. Again, anterior 2/3. Okay. Cranial nerve number 9, or glossopharyngeal nerve, posterior 1/3, and then the epiglottis and pharynx through the vagus nerve. Again, quickly, clinical correlations. Obviously, any local damage to the nerve. Okay, any local damage to the nerve, loss of taste, and you know this already. What is hyperacusis? Yes, good. Okay, older people suffer from this. Okay, where sounds that you kind of normally would not affect you in any way, humming off an AC, closing the door, that kind of thing. Again, this is due to paralysis of a muscle in the ear, okay? And the muscle doesn't hold on to it, so the thing vibrates, and therefore you have this thing. Okay, and then glossopharyngeal nerve, obviously, to the 1/3 at the back. Anything related to the nucleus solitarius, obviously, will cause ipsilateral loss because it's going to the same side. And then the thalamus will be contralateral loss because you're going to the other side. Ageusia means loss of taste. Hypo means lack, decrease, if you want, of taste. Sometimes we don't know. Happens in pregnancy a lot. People have cravings for all kinds of weird and wonderful pickles and so on. Medications, ACE inhibitors. Okay, ACE inhibitors, clotting agents, metronidazole, okay, antibiotics and so on and so forth. Zinc deficiency will also cause a decrease in taste. Bell's palsy, obviously, facial nerve to the face. And then COVID-19, we know that one. Dysgeusia, distorted taste. This is where people feel like the thing that's in their mouth tastes like a metal and so on and so forth, or like blood. Okay? And this happens with antibiotics and so on because they get secreted into the saliva. And it then triggers the taste buds and then you get this feeling. Okay? So, okay. Which brings me to smell, which is the other chemical basis of sensation or sensory input. Again, olfaction, it can tell you whether you are liking something or whether something, is not quite right. So if you picked up that shawarma that you left in the fridge last night and you kind of smelt it, you'll say, no, this needs to go into the bin, okay? Or that half-eaten yogurt or whatever it is. You understand what I'm getting at, okay? Okay, it can warn you of potential harm like spoiled meat and so on, or, if the house is burning down. Now, we can smell quite a few things, however, only 20%, only 1/5 of them are actually things we actually like, all right? Now, where do we smell these things, or where do we detect them? We detect them in the olfactory epithelium, right on the top of the nose, just above the superior conchi. It's about 10 centimeters square in human beings, and it's about 170 centimeters square in a dog. Obviously, any one of you have been to an airport line and the guy walks with the dog around. Obviously, if you go to Australia, the dog is not looking for drugs, it's actually looking for food. Okay? Probably they have one for drugs as well. But Australia is very strict about bringing in food into their country. Okay. So dogs have a significantly larger olfactory epithelium, therefore they can detect a lot more of these odorants, if you want. Again, there's the lady, and obviously you have to smell roses when you're drinking Saudi champagne, so the odorant will get into the nose, and this time you do not have a cell in between, you're going directly on to the olfactory neuron, and it's got these microvilli kind of branches to increase the surface area to detect the odorant. And therefore you will get, the minute you go over, and I'll tell you how you go over, the threshold you will get, the action potentials being triggered here, obviously to the brain. If you remove the stimulus, you don't detect the odorant. However, if the stimulus remains there for a long time, then this thing will also decrease. For example, if you use your particular perfume, you'll smell it when you put it on, but then, 10, 15 minutes later, you smell nothing. But you'll smell your friend's perfume when she walks by. Yes? So that's the kind of thing I'm talking about. It's called adaptation. Okay, let's look at where this olfactory epithelium is. There's it up here on the top. So you have the ethmoid sinus here in the front. You have the sphenoid sinus at the back. There's your pituitary gland and so on and so forth, your hypothalamus. And this little yellow area is what we call the olfactory bulb. So there's the epithelium here with all of the little branches, if you want. And it goes through this bone called the cibriform plate, and it makes this first cranial nerve or olfactory nerve, and it goes directly into the cranial area. And that's the one that obviously will not go through the thalamus. Okay, so cranial nerve number one, or olfactory nerve, does not go through the thalamus. Again, where does it go to? It goes into the olfactory bulb, and then you have tracts going into the cortex, or the sensory cortex, and that will go into the central nervous system like this here. Again, it's in the nose. Obviously, the nose also has things like mucus, okay? And there is, the reason they've drawn this purple kind of thing, there is a layer of mucus there that kind of concentrates the odorant molecules in and around these villi-like structures of the olfactory neurons. Again, location is obviously in the superior nasal area, just above the conchi. There's the inferior, medial, and superior conchi, and then the neurons will go into the brain. And then cranial nerve number one. Again, the pathway is through the olfactory receptor neurons, through the plate, and then into the olfactory bulb. Let's look at that one step further now. This little arrow, can you see this arrow here? What is it showing you? It's showing you this. So there's that epithelium, okay? And start up here, there's a cibriform plate. You have a lamina propria kind of thing between the plate and the epithelium, and there's the epithelium now, the olfactory epithelium. Okay, the epithelium has like about 3 or 4 different kinds of cells. So you have an olfactory gland that's essentially secreting the mucus. Then you have the actual olfactory neuron, and there's the cell body, so this will be the dendrite, and that would be the axon. Then you have the supporting cell, and then you have a cell here at the bottom, the basal cell, which can divide to form more olfactory neurons. So those are, the kind of cells that go along with this thing. Now, where is it? It's here on the next slide. So the basal epithelium will generally divide to give you more olfactory sensory neurons, okay? And again, that's the epithelium. The supporting cell, all right, is likely a glial cell that kind of produces mucus, and then you also have the olfactory gland that will secrete mucus as well. Why do you need the mucus? To kind of concentrate the odorant molecule. To concentrate the odorant molecule in and around these branches of the dendrites, so that you can smell this thing. Okay? And again, it regenerates. Okay, these olfactory receptor cells can regenerate every about 2 months or so. Okay, how do they work? So they work like this. Again, you need a chemical. The chemical attaches to a receptor on the cell that activates a G protein. Now, if you remember last year, Dr. Ahmed, G protein, GI, GS, this is a very specific G protein. It's not that one. It's just called GO. Okay? It's olfactory G protein if you want. And what does it do? Activates adenylate cyclase. You have cyclic AMP that opens up a cation channel, a sodium channel. Sodium comes in, you get depolarization, and now you can transmit. Okay? So, the chemical odorant will dissolve, become concentrated in the mucus. It's generally soluble, okay? And again, it will attach to these receptors on the cilia, all right? And it will activate the opening of this nucleotide opening channel, if you want, and then sodium will come in, and now you get activation, in that neuron. Looking at this another way, there's the cilia, and again, there's the odorant molecule, okay? And you got the olfactory kind of thing, cyclic AMP, opening up calcium and sodium channels. Again, that will then also open up a chloride channel that will make the inside of the cell even more positive. Therefore, you can smell this thing really well. Okay? And now you get depolarization, and that will go through the axon, through the cibriform plate, towards the olfactory bulb. Okay? Again, olfactory response can terminate because the cyclic AMP can do other things inside the cell. Okay, cyclic AMP may activate other signaling pathways, which then terminates the actual smell signal. Okay, if you are exposed to the odorant for an extreme period of time, the cell will then adapt to the odorant and not trigger the impulses, and that is what we would call adaptation. Okay, I gave you the example of the perfume kind of thing. It also applies, to go into Kentucky Fried Chicken and you can smell it from 10 miles away, and as you get closer to it, you don't smell the Kentucky Fried Chicken when you walk through the doors. You just smell the frying fat. Okay, which raises another question. If I have only 10 centimeters square of the olfactory epithelium, how is it that we can detect so many of these different kinds of smells? Now, if you are wondering about detecting these different kinds of smells, just ask the French, because they put out all these perfume kind of things, supposedly to entice us to buy it and so on and so forth. But they are also experts at wine. They smell it and it's supposedly fruity and God knows whatever and so on and so forth. So the question now is, how do we have so few receptors but we can detect so many different kinds of smells? And I've got the epithelium here. And the epithelium is represented by the olfactory neuron, and it's colored. Can you see it's colored?
Yes.
Good. Now, let's say somebody had a citrus kind of smelling, whether you are smelling lemons or oranges or, some perfume that has bergamot or patchouli in it or whatever the damn thing is. What will happen here? Just like taste now. Look at what's going to happen. So the green ones, notice they fire a lot. The blue ones a little bit. The red ones just increase their membrane potential, but no spikes. And remember, these are just action potentials going to your brain. When they go to your cortex, you then link that with citrus. Are you with me? Yes. In other words, you are linking a population of neurons in your brain with a particular smell. Okay? That's why it's called population coding. All right? On the other hand, you get the idea here. On the other hand, if you had a floral kind of thing, okay? And now you got some green, lots of blue, and maybe some red as well. That tells you that's a floral kind of smell. Okay? And, almond gives you the one at the bottom. So, again, each receptor will bind to a particular odorant. Okay? That will then trigger off these spikes, if you want. The outcome is that the odorant activates many of the 100 different types of receptors. Even not having anything to do with the receptor tells you it's almond. Okay? So this pattern of nothing, maybe a little bit of blue and a lot of red is almond. Okay? And that's how our brain then interprets that to say, this is what we are detecting. Which, again, I'm repeating the slide, I had it earlier on. So you're detecting, and now it's going to go to the brain. Again, the olfactory receptor neuron will trigger. Cranial nerve number 1 goes through the cibriform plate and goes into the olfactory bulb. Now what does it do in the olfactory bulb? It will form a synapse with groups of neurons called tuft or mitral cells. And then from there, you will have the postsynaptic neuron transmitting to the primary olfactory cortex. And it goes to various parts. Okay? It can go to a region called the uncus, which is in the temporal region in a little ridge on the side. Piriform. amygdala, notice a lot of this goes to the amygdala, the entorhinal cortex. These are limbic regions. In other words, learning, memory, like, you know. Emotions. Yes, emotions. That's what this is. All right? Again, there's, you know, it doesn't go through the thalamus. Okay. So if you're looking at the olfactory bulb, there's it. So cranial nerve number 1 will come in. It will synapse with these ganglion kind of thing called Tuft cells and mitral cells, and then they will project into the central nervous system. And then obviously amygdala, entorhinal cortex for learning, amygdala for emotions, and then piriform, and then from there you will go to the thalamus. Okay? Generally it goes from here up to the cortex, but now you got the other way around. So again, I put this little blurb in the corner here. When I was preparing the slide, you know how when you're on the internet, you get that little thing coming on the side, and I clicked on it, and supposedly women have a better sense of smell than men.
Absolutely.
Absolutely. Women have a better sense of smell than men, and women's sense of smell is stronger during ovulation, apparently. Okay? Again, when you get from the cortex, from, you know, the piriform cortex, the olfactory tubercle, it goes to the thalamus, then you get conscious determination, like I was saying, citrus, floral, and so on. That's when you will get conscious determination in the orbitofrontal cortex of the brain. Okay? There is this thing here, and it was in the diagram. I don't know what book I got it from, but in the diagram. And this is called the vomeronasal organ. And this works in animals. One second. This works in animals. It supposedly has not developed so well in, or has lost its development in human beings. What does it do? It detects pheromones. Now, the example I gave you is the animal weeing on the tree or rubbing its body on the tree, and then the other male comes in or the female comes in, whatever it is. And that's what is used here, this vomeronasal organ. Okay? And it comes in through the nose, obviously, and you have this accessory olfactory bulb somewhere up there. Again, for humans, they don't know. Okay? Now, it's perhaps, you know, human beings may have evolved differently. These days, we kind of go out to dinner, and we share the meal, and we talk, apparently. And we decide whether we like what, you know, the common things, and so on and so forth. Okay, and we ask questions, you know, like how much money you're going to earn from your great-grandfather's death, and so on and so forth. And that is what determines whether or not, you know, there's compatibility between individuals, rather than going around smelling trees. So that's what that is.
Do we have this structure as a comment on this?
No. People have been trying to find this thing for ages, and they haven't found it. There was a study done in the 70s among women who lived in a dorm, okay, and they had all of these women living in the dorm, and then they found that among the women, some of the women went into their menstrual cycle, into their period, and then as they remained in the dorm, it's almost like they synchronized their menstrual period. There's no truth to the story. I mean, the story is published, but it's not scientifically proven yet.
I told you.
I lost track. Okay. So this diagram, I put this diagram obviously in words. So again, olfactory bulb, and then, you know, you're going to go track into the brain. Again, no thalamic relay, and then, you know, and I wrote that for you again in words. Okay, olfactory dysfunction now. Because the olfactory, the cranial nerve number 1 goes through the cubiform plate. Any shearing of the cranial nerve for any reason, in other words, damage, car accident, whatever it is, you're going to have anosmia. Now, if you remember Kalman syndrome from last year in reproduction, okay, you get anosmia again with hypogonadotropic hypogonadism, if you want. Again, Parkinson's disease for some reason or the other. Long before you start getting the shaking and so on and so forth, there is an early loss of smell as well. Okay? The word prodromal means long before. It's not a total loss of smell, but it starts decreasing. Okay? And again, it might be because of these precipitations or Lewy bodies that you find in the neurons. Alzheimer's disease causes hyperosmia. Okay, again, this might be because of the buildup of those fibrillary triangle, and the tau proteins and all that story. COVID-19, we all went through that a couple of years ago. Zinc, for some reason or the other, is required to make that olfactory epithelium, to regenerate it. So when you don't have sufficient zinc, that is affected. And then lastly, obviously chronic inflammation of the olfactory epithelium will cause this problem as well. I think that's enough for me. Thank you.
Thank you, doctor.